it sounds like there's some progress and some confusion, which is okay. I like to talk about these experiments because I think it's fun for us to like look back and imagine how they posed questions and how they designed experiments to answer those questions using pretty simple technology, really. I mean, there's nothing here more than a glorified washing machine and some salt solution. It's slightly more complicated, but basically that's it, right? Something you could do at home. So let's imagine we're a bacteria, and we are growing in heavy nitrogen DNA. I'm sorry, we're growing in heavy nitrogen, and this is the DNA we've synthesized. All right. If we're a bacteria growing in normal nitrogen, nitrogen 14, this is what our DNA looks like. So blue versus black. Okay? So if you take and centrifuge light nitrogen DNA, so this, sorry, the normal nitrogen, all of that DNA only goes down a little ways in the tube because it's not very dense. So that double helix would be like right here. Okay? And if you take heavy nitrogen and you do the same thing, well, all those DNA molecules are much denser, and so they move all the way down to there. So if you take bacteria that is all heavy and you let it use the building blocks to make new DNA that's all light for one round of copying, okay, what you end up with is, so this is going to be after 20 minutes, which equals one round of copying, you end up with DNA that all occurs right here. What do you think that DNA looks like using this black and blue approach that we're talking about here? Or better yet, what doesn't it look like? Which of the possibilities can we conclusively exclude at this point?
Dispersive.
You can't exclude dispersive. It depends on your model for dispersive. The way they modeled it up here, you wouldn't be able to exclude it. Here. Because it would still be half heavy and half light. It kind of depends on, there's a number of ways that people describe dispersive in terms of the makeup of the strands. But the way they've described it here, I think you can see it's half heavy, half light. Now, somebody said you can exclude conservative. And do they want to explain why or how?
Yes.
Go for it.
So the reason why they have to exclude conservative is because conservative is represented all the way on the left by the heavy nDNA previous to them replicating with light nitrogen isotopes.
This one?
Yeah, and that's previous to the 20 minutes and 40 minutes examples where they basically go through reproduction with the light nitrogen isotopes. And so if it were to be conservative, they would be conserving all of the nitrogen, the heavy nitrogen isotopes from before they went through the reproduction process. And so if you go through one single 20-minute process, you will find two different lines, the heavy being all the way on the bottom or just staying all the way on the bottom.
Yeah. Exactly right. And so I think the best way to conceive of this, or to think about the results, is to think what the results would be if it were one of these cases. So if it is conservative, what you'd have at the end is you would take your heavy DNA, you'd separate the two strands, you'd make your complementary copies, and then you'd reassemble the heavy and assemble the light together. So what you would have ended up with if it were conservative, don't get confused here. I'm going to write up what it would have looked like if it were conservative, is you would have had DNA down here and DNA up here. But we didn't see that. We didn't see any DNA up there or down at the bottom. We only saw DNA right here, and that's because, well, it's consistent with it being semi-conservative. Where you separate the two heavy strands, you synthesize a complementary light copy, and you're done. Now imagine what would happen if you took this one more round of replication. What would happen if you took DNA that looked like this and let it copy itself one more time using light materials? What would you end up with? What's that? Okay, so the new DNA would be lighter, and how light? Would it be as light, or would it be somewhere in between? It would be as light as pure light DNA. Because think about what would happen if you took this strand and you separated it. I'm going to draw it over here briefly. So here's our double strand. Remember, the black is the heavy, the blue is the light. I'm going to separate these strands. And now I'm going to synthesize a new complementary copy using only light materials, which are blue. So what would that look like when you centrifuge it?
2 layers. This
Half heavy, half light would migrate to exactly the same place as it did in the previous centrifuge. You guys see that? And the new one here, which is all light, would migrate to the same place as the all light one did. Whoa, I missed that. Let's try to be consistent here. There we go. It's kind of a cool experiment, isn't it? I think it's a neat, simple, elegant experiment that figured out something very basic and fundamental about how living systems copy heritable material. Does anyone have any questions about these results? Yeah.
How recent was this experiment?
This was 60s, 50s maybe, late 50s,
Late 40s, early 50s.
something like that.
Okay.
Yeah. So it was right around the time of the discovery of the double helix. It was in that same general vicinity. So yeah, it's not a super modern discovery, but it is a pretty cool experiment.
That's cool. And then, like, so going back in the petri dish, they have basically just 2 colonies of bacteria, one with the light nitrogenous bases, one with the heavy nitrogenous bases.
Yeah.
And then when they marry the two, it's the anticipation that those, as they progress, are taking each other's DNA.
It's not that they're taking each other's DNA, it's that you shift the bacteria out of a medium, like out of a broth that has heavy nitrogen precursors for DNA into a broth that has light nitrogen precursors.
Okay.
And so as they reach out into the environment to grab what they need to build their new body, they grab the light ones because that's all around. That's all that's around.
So it's not 2 colonies, it's just 1 colony in different broth.
Yes.
Okay.
The truth is they wouldn't be growing it probably on petri dishes. They'd be growing it like in flasks in solution, but I didn't know if that would be as universal a symbol for bacterial growth as. Okay. Any other questions about this experiment? So keep in mind these experiments that we went through over the last day and a half. The Avery, the Griffith, the Hershey-Chase, and now this Michelson-Stahl experiment. Because I might ask you to explain one of them on a midterm. Or maybe all 4. Why not?